Hello, welcome to my channel. So today we are going to talk about the muscle tissue. So this is also another human anatomy and physiology lecture. So let's go straight into the lesson. So our objective for today, of course, are going to describe the general features of muscular tissue. We are also going to contrast the structure, location, and mode of action of the skeletal, cardiac, and smooth muscle tissue. Okay, then you have to know that muscular tissue consists of elongated cells which are known as muscle fibers, okay, or myocytes that can use ATP to generate force. As a result, muscular tissue produces body movements, maintains posture, and generates heat. It also provides protection. Okay, then you also know that the muscle tissue is mesodermal in origin, and it is named based on its location and certain structure and functional features. So we have in terms of naming of muscles, so location, certain structure, functional feature, and also the muscular tissue is classified into three types. Where we have the skeletal, cardiac, and smoother muscles. Okay, so function of muscle tissue, of course, is for movement, maintenance of posture, joint stabilization, heat generation as well. Then we have other special features of muscle tissue, where we have contractility. So for contractility, only one action, to which of course to shorten, and shortening generates pulling force. Then for excitability, the nerve fibers cause electrical impulse to travel. Then for extensibility, they stretch with contraction of an opposing muscle. Okay. Then elasticity, they recoils passively after being stretched. That's some special features of muscle tissue. Then for special terms that you have to know, we have plasma membrane. So the plasma membrane for muscles, we call it sarcolemma. Then the cytoplasm of muscles, we call it sarcoplasm. Then in the smooth endoplasmic reticulum for muscles, we call it the sarcoplasmic reticulum. Then the mitochondria of muscles, we call it sarcosomes. That's some of the things you have to know. So muscle tissue is made up of basically of cells called myocytes. Then myocytes are elongated in one direction. So these are an example of myocytes. Okay, so and together they make up the muscle fibers. Okay, so the muscle types we have talked. They have skeletal, cardiac, and smooth muscle. Okay, and they are cross-sectional values that you have to know, and their activity. Okay, so in terms of cross-sectional, or in terms of activity, skeletal muscle are strong, quick, discontinuous voluntary contraction. Then cardiac muscles are strong, quick, continuous involuntary contraction. Then the smooth muscle also have a weak, strong involuntary contraction. Okay, but let's now just talk about them in detail specific. Okay, so starting with skeletal muscle tissue, the description, they are long, cylindrical, striated fibers with many peripherally located nuclei, and of course they are of voluntary control. Okay, so that's two features you have to know about skeletal muscles. Location, they are usually attached to bones by tendons. Then function, of course, for skeletal muscles is for motion, posture, heat production, and protection. That are some of the functions. Okay, then this is how a skeletal muscle looks like in terms of a longitudinal section. Okay, so what are the organization of the skeletal muscle? So the thin layer of connective tissue surround and organize the contractile fibers. Then you have the epimysium, which is the external sheath of dense connective tissue that surround the entire muscle, and the septa of the epimysium extend inward, carrying neurovascular bundle. Then you have the perimysium, which is a thin connective tissue layer that immediately surrounds each bundle of muscle fibers called fascicle. Then you have the endomysium, which is within the fascicles. There is a very thin, delicate layer of reticular fibers that surround external lamina of individual muscle fibers. Okay, so this is what we are talking about: the endomysium, endomysium, or perimysium, and epimysium. This is a structure of how it looks like. Okay, so the endomysium covers each specific inside, then the epimysium covers the whole bundle in all as a whole. Okay, then going forward now, so the skeletal muscle or the fibers each is one cell and have striations, of course. That's the skeletal muscles, one of the features that they have. And the myofibrils are organelles of the cell, so these are made up of filaments. Then the sarcomere, which is the basic unit of contraction. The myofibrils are long rows of repeating sarcomeres. Then the boundaries which they have are known as Z-disc or lines. Okay, that's a demonstration of a filament. So there is a thin and a thick filament. So the actin and myosin filament. Okay, which has the H zone, we have the Z discs, and we have the A bands, and the I bands, and the M line. Okay, so that's so that's about the skeletal muscle. And then this is known as the how movement is initiated and muscles following ATP usage. Okay, so first things first, we are going to, of course, start off. Let me just zoom in. Okay, so after zooming in, okay, let me zoom back. Okay, so let me just zoom back so that people will be able to read. Okay, so you have to know that the nerve impulse triggers the release of acetylcholine. Okay, so it triggers the release of acetylcholine from the synaptic knob into the synaptic cleft. Then the acetylcholine is going to bind the acetylcholine receptors in the motor end of the plate of the neuromuscular junction, inhibiting a muscle impulse in the sarcolemma of the muscle fiber. That's one. So as you can depict, we are talking about at this level at the neuromuscular junction level. Okay, then after the neuromuscular junction level, as the muscle impulse spreads quickly from the sarcolemma along the teeth tubules, calcium ions are released from the terminal cystine into the sarcoplasm. So now at this point, then after that, the calcium ion is going to bind to troponin, and troponin changes shape, moving tropomyosin on the actin to expose active sites on actin molecules of thin filaments. Then the myosin head of thick filaments attached to exposed active sites to form cross bridges. Okay, that's 3. So we're talking about 3 here. So that's the actin and the tropomyosin. Okay, then 4, the myosin. So the myosin aids now pivot, moving thin filaments toward the sarcomere center, and the ATP which binds myosin heads is then broken down into ADP and P. Then the myosin heads detached from thin filaments and return to their pre-pivot position, Then the repeating cycle of attached pivot detach returns slides thick and thin filaments past one another. So the sarcomeres shorten and the muscles contract. Then the muscle continues as long as calcium ions remain bound to troponin to keep active sites exposed. Okay. Then 5. So when the impulse stops, calcium are actively transported into the sarcoplasmic reticulum. Then tropomyosin recovers active sites and filaments passively slide back to their relaxed state. So this is about the from the neuromuscular junction up to the point where the impulse is going to stop. Okay, so this is particularly explaining about how movement is made, but specifically we need to talk about the sliding filament model, which again has been partly been covered. Okay, so number 1, under the sliding filament, you have to know that the sarcomere shortens because actin pulled towards its middle is pulled towards its middle by myosin cross where the myosin cross bridges. Okay, that's under. So we have the H band and the A band. So when the sarcomere shortens, because the actin is pulled towards its middle by myosin cross bridges. That's what's going to happen. So it's going to be pulled towards its middle. Okay, that's under the in a relaxed sarcomere. Okay, so when it is fully contracted, the A band, when it is fully contracted, that's it looks like this. So down here, so that's how it's going to look like. Let me try to zoom in. Okay, that's when it is fully contracted. Okay, so when it is fully contracted, as you can see, the A band is now the one which is now going to become more visible, and the H band is not necessarily going to be very visible. Okay, then the I bands are going to cover up most of the bigger spaces. That's under the fully contracted one. Then the partially contracted one, the A band, because it is caused by myosin, which doesn't change length, the under the partly contracted, even the A band does not necessarily change the length because it is caused by myosin, of course, and does not change the length.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When the muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T-tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay, so let's continue from where we ended. Okay, so the neuromuscular junction, as we talked about previously, so we have the motor neurons which innervate muscle fibers, and the motor end plate where they meet, and the neuromuscular transmitters which are released by the nerve signals which initiates the calcium ion release and muscle contraction. Then we have the motor unit, which is the motor neuron and all muscle fibers that it innervates. So these all contract together. The average is about 150, but the range is 4 to several hundreds muscle fibers in a motor unit. Then the finer the movement, the fewer muscle fiber or motor unit. So the fibers that are spread throughout the muscle, so stimulation of a single motor unit causes a weak contraction of the entire muscle. Okay, so then the type of muscle, skeletal muscle fibers, where they have the fast, slow, and intermediate. So whether or not they predominantly use oxygen to produce ATP, the energy molecule used in muscle contraction, that's the ATP, the same ATP. Okay, then we have, let's now talk about the fast fibers. So fast fibers, also known as what fibers? These are large, predominantly anaerobic, fatigue rapidly, meaning they rely on glycogen reserves, and most of the skeletal muscle fibers are fast. Then you have the slow fibers, which are red fibers and have the diameter 3 times slower, but can continue contracting. Then they are aerobic and more mitochondria, and they have myoglobin as well. Then the intermediate one, they are four in between the fast and the slow ones. Okay, so then you have to know that a skeletal muscle contracts when its motor unit are stimulated. The amount of tension depends on the frequency of stimulation, the number of motor units involved, and of course there is a single momentary contraction which is called the muscle So when there is a single momentary contraction, it's going to be called a muscle twitch. Then there is what we call O or none principle. So each muscle fiber either contracts completely or not at all. That's known as the O or none principle. And the amount of force depends on how many motor units are activated. Then the muscle tone. So in terms of muscle tone, so even at rest, some motor units are active, then tense the muscle even though not causing movement. This is known as the resting tone. Okay. Then some applied anatomy, or some applied anatomy to be specific, there's what we call muscle hypertrophy. So for instance, when weight training, where there is repeated intense workouts, increases diameter and strength of fast muscle fibers by increasing production of mitochondria in the sarcosomes. Then the actin and myosin protein production also increases. Then the myofilament containing these contractile proteins also increases, and the myofibrillar organelles, these myofilaments form. Then the other thing is the fiber is going to enlarge, that's the hypertrophy, as the size of the myofibrils increase. So the muscle fibers or the muscle cells don't increase in number but increase in diameter, producing larger muscles. Okay, that's about hypertrophy. So hypertrophy is more of just an increase in size. Then hyperplasia is an increase in the number of muscle cells due to increase in number of muscle fibers. Then muscle atrophy is the loss of tone and mass from lack of stimulation. So muscles become smaller and weaker due to lack of stimulation. So we are done on skeletal muscles. So let's now talk about the cardiac muscle tissue. So the cardiac muscle tissue is a branched striated fibers with one or two centrally located nuclei and contains intercalated discs, and they are of involuntary control. So intercalated discs striated, and with one or two centrally located nuclei. That's the description. Okay, then location is the heart wall. Then function pumps blood to all parts of the body. This is the longitudinal section of the cardiac muscle tissue. Then you have the smooth muscle tissue, whose description, of course, is that they contain the spindle-shaped, which is the thickest in middle and tapering at both ends. They are non-striated fibers with one centrally located nucleus, and they are also involuntary control. Then the location, they are found in the iris of the eyes, walls of the oral internal structures such as blood vessels, airways to the lungs, stomach, intestines, gallbladder, urinary bladder, and the uterus. That's the location of the smooth muscle. Then function, they act for motion, such as in constriction of blood vessels and airways, and also for propulsions of foods through the gastrointestinal tract, and contraction of the urinary bladder and the gallbladder. Okay, this is also another longitudinal section of the smooth muscle tissue from the arteries. Okay, then there is what we call regeneration of muscle tissue. So under regeneration of muscle tissue, you have to know that skeletal muscles show a limited regenerative capacity, and the satellite cells living within the skeletal muscle are implicated in regeneration of the skeletal muscles. In cardiac muscles lack satellite cells and show very little regenerative capacity beyond early childhood. So damage to the heart is replaced by a connective tissue. Then for the smooth muscle, smooth muscle is capable of regeneration and can undergo mitosis and replace damaged cells. Okay, so these are some of the differences between the skeletal and smooth and cardiac muscle fibers. So you can pause and go through the differences. Okay, because this is an examinable question where you ask about the differences. So please pause and go through. So that's the end of the tutorial. Then these are just a checkpoint. You have to understand which types of muscular tissue are striated, which are smooth, and which type of muscular tissue have a gap junction. And that's all. Thank you for watching. If you haven't subscribed, please subscribe and hit the bell notification. And see you on the other tutorials on the human anatomy and physiology lecture. Thank you once again.